The following is a selected video from masterthecontent.com, where you will find an extensive video library of lectures for a variety of standardized admission tests. We offer over 600 hours of detailed video lectures for a multitude of standardized tests. Use our interactive in-lecture table of contents to find specific topics of interest. Work through numerous in-lecture examples to help you internalize concepts. To learn more, visit masterthecontent.com. Your career, our passion.
We are going to talk about is gene expression. So we're going to try and define this term in a very general sense. So what is gene expression? It's a process by which DNA directs the synthesis of proteins. A very simple statement, very concise, very precise. It's basically the process by which DNA will direct the manufacture or the synthesis of proteins in a cell. Now, it's important to note that DNA does not actually direct the synthesis of proteins directly. It's not directly involved in the formation of these proteins. It actually delegates this task. So genes will actually provide instructions in the form of what we call an RNA transcript. And those instructions are what then are used to synthesize a protein. Now, what you can think of RNA or ribonucleic acid as being, in this case, is therefore a bridge between the genes in your DNA and the proteins that they encode. Now, there's a few important things I should mention about RNA, some of which you should already know. Now, RNA obviously is very chemically similar to DNA. The only difference is that it contains a ribose sugar instead of a deoxyribose sugar, which you will find in DNA. It also has the nitrogenous base uracil in place of thymine. And typically, you're going to find RNA molecules existing as single-stranded molecules, a single strand, as opposed to DNA, which usually occurs in double-stranded fashion. Now, the building blocks of both DNA, RNA, and RNA are nucleotides. You should remember what a nucleotide is. A nucleotide is basically the monomeric unit, or the smallest unit of structure in DNA and RNA. So what a nucleotide consists of is a nitrogenous base, a 5-carbon sugar, and a phosphate group. So we actually examined nucleotides at length in a previous lecture. If that's information that's a little hazy to you at this point in time, you might want to revisit that lecture and refresh your memory as far as nucleotide structure is concerned. So, how then are genes encoded in the structure of DNA? So genes are actually encoded in the specific sequence of nucleotides in DNA. So a gene will typically be hundreds to maybe thousands of nucleotides long, and each of those genes will actually code or encode a very specific set or sequence of amino acids in a polypeptide chain. So what we're interested in knowing in this case is how do we move from a gene to a protein? How do we get proteins from genes? Now, getting a protein from a gene requires two main processes. The first one being, I already talked about it very briefly, the first one being transcription, and the second one being translation. So transcription is the first step, the very initial step in gene expression. And what happens in transcription is that a DNA segment is used as a template and is converted into a complementary segment of mRNA. So you take a specific segment of DNA and you convert that segment of DNA, which is a gene, into a specific sequence of nucleotides in what we call rRNA, ribonucleic acid. Now this RNA, this ribonucleic acid, is what then we will use to synthesize our protein. So as you can see, the genes are encoding proteins indirectly. We're copying information from the gene, and then we're using that information copy in the form of RNA to direct or to control the synthesis of a protein. Now, the second step in, the second most general step in the synthesis of proteins or in gene expression would be translation. So I've already said that RNA will direct the synthesis of proteins in the process of translation. So what translation is, or what the definition of translation is, it's the process in which RNA produced during transcription is read or decoded by a ribosome to produce a specific sequence of amino acids. So I've already talked about a number of different important molecular structures that you should have in mind by now. I talked about genes. So what a gene is, it's a specific sequence of nucleotides in the DNA molecule. So what can happen is that we can take that sequence, we can transcribe it in the process of transcription and create an RNA copy, complementary RNA copy of that sequence. We'll then take that RNA copy and we'll use a host of different other molecular aids and proteins such as ribosomes and enzymes to then use or then to then synthesize or make a polypeptide chain or a protein. So what happens when your cell needs a particular protein? The nucleotide sequence is copied from DNA, and then that nucleotide sequence is converted into RNA, and then that RNA is what is then used as a set of instructions to dictate or to guide the formation of a protein based on that gene, based on the nucleotide sequence of the original gene. So this flow of information in one direction, this unidirectional flow of information from DNA to RNA and from RNA to proteins, it's a fundamental principle of biology, and it's what we call the central dogma of molecular biology. So that's what we call the central dogma of molecular biology. And all it seeks to explain is the transfer of genetic information in biological systems. And in this case, we're talking about the movement from a gene to RNA and from RNA to a protein. Now, all cells of all organisms, including bacterial cells, or for instance, even eukaryotic cells like our cells, express genetic information in the same way. So we all use this same set of mechanisms, albeit slightly different in one way or the other, but we all transcribe our genes and we all translate those transcripts to produce proteins. Now, what I want to do is I want to move on and I want to talk about or continue talking about the movement from a gene to a protein. Now, it's important to note that DNA serves as a template in two separate scenarios. So it can actually serve as a template for the synthesis of a new strand, and I'm talking about replication. We covered replication in a previous lecture, and it can also function also as a template for the assembly of RNA or an RNA transcript during the process of transcription. So DNA serves as a template, one, in replication when you're looking at doubling the genetic material in a cell, for instance, for cell division, and it can also function in replication when you're interested in extracting or pulling out specific genes from DNA and using those genes to direct the synthesis of proteins. Now, transcription, however, is significantly different from replication, although both those processes use DNA as a template. So unlike a newly formed DNA strand in the process of replication, an RNA strand does not remain attached to the DNA, so it's released as it's synthesized. Okay, so, and because only one strand of DNA is transcribed, only one strand of RNA is typically produced. And another difference is that the RNA that is copied from a limited region of DNA tends to be a lot shorter than those DNA segments. So RNA segments in general, not always, but in general tend to be a lot shorter than their counterparts and than DNA segments. So the RNA that is produced in the process of transcription has a special name. It's called mRNA or messenger RNA. So why do we call it messenger RNA? We call it messenger RNA because it carries a genetic message that contains the protein building instructions for a particular gene. So it contains a genetic message which is used to build a protein which is dictated by a particular gene. Now, the actual physical process of translation occurs outside the nucleus. It happens in a structure we call the ribosome, and we will look at the process or the mechanism of translation in much detail later on in this lecture. But I want to mention a few additional important things about transcription and translation as far as eukaryotes and prokaryotes are concerned. Okay? Now, a ribosome is the site at which mRNA is translated. So it forms a site at which we will read that mRNA and convert that sequence of nucleotides and mRNA to a complementary sequence of amino acids. Now, this process is universal, like I said, in that it occurs in all organisms. But one important distinction that I want you to make is that of compartmentalization. And I'm talking about a difference between eukaryotic cells and prokaryotic cells. Now, because of the fact that bacterial cells do not have a nucleus, their DNA is not separated from the general cell cytoplasm. What does this mean? This means that as genetic information or genes are being transcribed, they can also be simultaneously translated to produce a protein. So there's no separation of these two processes when we're talking about bacterial cells, when we're talking about prokaryotes. Eukaryotes, on the flip side, on the other hand, actually have distinct compartmentalization of these two processes. Okay, so transcription in a eukaryotic cell is separated physically from translation. So what happens is that transcription will occur in the nucleus of a eukaryotic cell, and then that mRNA transcript, that messenger RNA that is produced as a result of that transcription process, is what is then transported outside the nucleus, where it then is used in translation to produce a protein. Now, another important distinction that I need to talk about before I move on is the processing of transcripts. So typically in prokaryotic cells, the transcription product, or the mRNA transcript, is ready for use. As soon as it forms, it can be used as a template to direct the synthesis of a polypeptide chain, to direct the formation of a protein. Now, eukaryotes, on the other hand, have somewhat of a different situation going on. The initial transcript that is formed from transcription, we call this the primary transcript. You can also call this pre-mRNA, is immature. It's not ready. It's not ready to be used for the synthesis of proteins yet. It must undergo additional processing before it can then be removed from the nucleus to perform its function. So that's another key distinguishing feature between the transcription and translation processes when we're talking about prokaryotes versus eukaryotes. So I'm just going to move on.